Welcome to PW English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you to this lecture in the UMEED NEET English batch. Today we're going to discuss the chapter Respiration in Plants in absolute detail in one shot. Before we get started with the discussion of the concepts, let me take you through the analysis of how many questions have been appearing from this chapter over the last few years in NEET. As you can clearly see, there are around 1 to 2 questions roughly that have been asked from this chapter every year. Most students find this chapter very difficult to understand, but hopefully after this session, it will all be very easy to you. Okay? So, what about the syllabus then? Is there any change in the old NCERT and new NCERT? For this chapter, I would recommend you to refer the new NCERT, not because there are some concepts that are deleted. No concepts have been deleted from this chapter. Whatever was there in the old NCERT is still there in the new NCERT. But in certain concepts, the content has been updated. Certain, sentences have been updated and changed. So the latest NCERT is what I would recommend you to refer for this chapter. Okay, so let's get started. What is respiration? The moment you hear the term respiration, what is the first thing that comes to your mind? Most people generally associate or equate respiration with breathing. Breathing is taking in air, giving out air, right? You take in oxygen, give out carbon dioxide. That physical process of taking air into your lungs and releasing the air from your lungs is what we call breathing. Breathing itself is not equivalent to respiration. Breathing is a physical process. Respiration is something that happens inside of our cells. So we call it cellular respiration. I'm not sure if you remember or not, but when we discussed the chapter, The Living World, I told you how cellular respiration is a characteristic feature of all living organisms. From the tiniest bacteria to protozoans, plants, animals, every living organism does undergo cellular respiration, right? Though this chapter is called Respiration in Plants and we're learning about how it happens in plants, very similarly, it will happen in all living organisms also. The reason why we don't consider viruses as living organisms entirely is because they lack this process of cellular respiration. That's one of the reasons why we don't consider them to be living, right? So, respiration is a process that includes breathing in some organisms, but respiration itself is not the same as breathing. I want you to keep that in mind first. All right. So, what exactly does respiration do? When you think about the things that you absolutely can't live without, we cannot live without, is food and oxygen. You can probably live without food for a couple of hours, days, but you cannot live without oxygen beyond a few minutes, right? Everybody knows why food is important. Food is important because that gives us energy. Why do we need oxygen at all? Why does our body need oxygen? What does it do with all the oxygen that we breathe in? Why is oxygen essential? Oxygen is what is required to get energy from the food we eat. All right? So, regardless of what you've eaten, it could be chapati, rice, whatever. Once you chew it, once it goes into your alimentary canal, in different parts of your alimentary canal, different enzymes will, act on it and it will break it down into smaller molecules. What happens to those smaller molecules? These smaller molecules, the smallest monomer units of whatever food you've eaten, gets absorbed from your small intestine into your bloodstream. And your heart will pump this blood to every one of your living cells, right? So the food that we've eaten gets broken down in our digestive system. The circulatory system will carry the nutrients that we get from food and deliver it to every one of our cells. Why do our cells need nutrients? Because our cells will use that broken down food as energy, right? If you think about us at the moment right now, I'm talking, you're listening, you're probably watching and taking notes, right? So while I'm talking, I'm moving my jaw, I'm moving my tongue. So movement of jaw, movement of tongue requires energy. You know how muscle contraction consumes energy, right? It requires energy. You've learnt about it in locomotion and movement, right? Actin, myosin sliding past each other, that consumes energy in the form of ATP. So if I have to talk, I need energy. If my heart has to pump blood, it requires energy for contraction, right? And in our cells, there are a lot of metabolic reactions that consume energy. If proteins have to be synthesized, that requires energy. If carbohydrates, polysaccharides have to be synthesized, glycogen has to be produced, that requires energy. So a lot of metabolic activities that take place within our body require energy, and that energy we get from food. So whatever food we've eaten, that gets broken down, gets supplied to all of our cells, and within our cells oxygen is used to break that food down and release energy. That energy is utilized by our cells to perform its metabolic activities, right? Like right now, if you're looking, then the information has to be transferred from your eye to your brain. And if you're listening to something, information has to be transferred from your ear through your auditory nerve to your brain. So a lot of this requires energy, and that energy we get from food. How is food converted to energy? When food gets oxidized, energy is produced. One of the ways food gets oxidized is by using oxygen. Okay, so oxygen is utilized for the oxidation of food in most organisms, and that process releases energy, and that energy is what we utilize for our metabolic functions, right? So yeah, basically life processes require energy, and we obtain that from oxidation of some macromolecules. What are these macromolecules? These are our carbohydrates, proteins, fats, etc. that's present in our food. So when those get oxidized, energy will be released. Now, if you think about how different living organisms obtain energy, we'll talk about plants or autotrophs first. Autotrophs include our plants which are photosynthetic and also cyanobacteria which can perform photosynthesis, that is our blue-green algae, right? So in those organisms, they have the ability to trap the energy present in sunlight and utilize that energy for the formation of glucose. How exactly this whole thing happens, we've learned in our previous chapter, right? So by using energy in the sunlight, they have figured a way to convert it into food. They are self-sufficient. Whatever energy they require, for that whatever food is required, they themselves have the ability to synthesize that food. But think about a plant. Take one plant for example, any angiosperm plant, alright, or any plant for that matter. Every part of the plant is not capable of synthesizing food, right? Whichever part of the plant has chloroplast, has chlorophyll, only over there photosynthesis can take place. You can find chlorophyll and chloroplast mostly in the green parts of the plant, which is the leaves. In young stems which are very flexible and soft, the stem also can sometimes be green in color. But the roots are almost always non-green, and they don't perform photosynthesis. Though we call the plant as a whole as autotrophic, every part of the plant is not capable of performing photosynthesis, right? The cells in the root cannot perform photosynthesis. So whatever photosynthesis the leaf has performed, the food that it has prepared there, it will have to be transported to the parts where photosynthesis does not take place, right? So wherever food, whichever part cannot perform photosynthesis, it receives food from the leaves usually, okay? So that is one thing about autotrophs. Talking about heterotrophs, we can classify heterotrophs into two categories. One is consumers like us. We eat plants, we feed on plants, and we are dependent on plants for food, either directly or indirectly. We ourselves lack the ability to synthesize food. Talking about decomposers or saprophytes like bacteria and fungi, many bacteria and fungi, they don't eat food like we do, they don't eat it, but instead they will depend on dead and decaying plant and animal matter. They obtain their nourishment from that. We eat plants that are alive. We kill plants and eat plants, or directly if you are vegetarian, or if you eat meat, then the animal that you're eating would have consumed some plants, right? So directly or indirectly, we are dependent. Consumers are dependent on autotrophs for our nutrition. Whereas bacteria, fungi, saprophytes, they also depend on other organisms, but they won't be live. They are dependent on dead and decaying matter. If a plant dies, it is utilizing the organic matter present in the plant that the plant got because of photosynthesis. If an animal dies, the bacteria and fungi will decompose and get nourishment from the organic matter that animal had, which also was in a way directly or indirectly from photosynthesis from plants, right? So ultimately, most majority of living organisms will depend on autotrophs for their nourishment. All right. So only green plants and cyanobacteria can perform photosynthesis. In photosynthesis, light energy is trapped and converted into chemical energy. I told you that. And that is stored in the bonds of molecules like carbohydrates, such as glucose, sucrose, starch. Whichever cell contains chloroplast, only that cell can perform photosynthesis. Even in plants, other organs that are not green, non-green, such as root, like I mentioned, they need food for oxidation, which they will get from leaves. All right, it will be translocated from the leaves. Animals are heterotrophic. They obtain food from plants either directly or indirectly. Saprophytes, such as fungi and bacteria, they also obtain food or nourishment, nutrition from dead and decaying matter, which ultimately came from plants itself from photosynthesis, right? So this is the introduction. Now, whenever cellular respiration has to happen, it will happen inside the cells. Inside every living cell, it will take place. Some steps or some stages of cellular respiration happen in the cytoplasm. Some can happen inside organelles like mitochondria in case of eukaryotes. But if it's prokaryotes, some steps will happen in the cytoplasm and some steps will happen in specialized structures called mesosomes, about which I've told you and explained in the chapter Biological Classification. I hope you remember. I told you how during respiration there are some macromolecules that will get oxidized. These macromolecules could be carbohydrates, proteins, fats, organic acids, etc. And that will get oxidized. And when those macromolecules get oxidized, we obtain energy, right? That's what I told you. So there will be certain macromolecules such as, carbohydrates, proteins, fats, organic acids. All of these obviously are organic compounds, so they will have carbon-carbon bonds. There is energy present in these molecules within those bonds. Whenever those bonds are broken, energy will get released. Remember, photosynthesis, we learned, I told you that photosynthesis is an anabolic process. It's a building up reaction where small molecules of carbon dioxide, 6 small molecules of carbon dioxide are coming together to form one big molecule. Whenever something is getting built up in nature, it will consume energy. It will require energy. For photosynthesis, that energy came from sunlight. For respiration, during respiration, what happens is the exact opposite. This glucose that has been prepared during photosynthesis can either be stored as starch in plants, stored as glycogen in us animals. Regardless, it is an organic molecule that contains a lot of carbon-carbon bonds. The energy is stored in these carbon-carbon bonds. Whenever this molecule gets broken down, the carbon-carbon bonds get broken, considerable amount of energy is released. That is what the cell will make use of to perform its energy consuming metabolic reactions. Alright, so yeah, so these macromolecules that I spoke about and I've listed right here, they have a lot of these carbon-carbon bonds. When they are oxidized, energy will get released. So whichever substances we are using for oxidation to release energy, now these are called respiratory substrates. In most cases, the respiratory substrate that the cells will prefer is carbohydrates. This is in most cases. Only when there is not sufficient carbohydrates or under other special conditions, proteins, fats and organic acids are preferred by the cell. Okay, so this happens under certain conditions only. But mostly organisms and cells prefer to use carbohydrates or glucose as the substrate for respiration. Okay, so these are respiratory substrates. Now, how is this energy stored? I told you that carbon-carbon, in between carbon and carbon, you have high energy bonds, right? The energy is stored in the molecule within those bonds. I also told you whenever something is getting broken down inside a cell, energy will get released. What basically happens during cellular respiration is, see, right now you may have eaten food. The food that you've eaten will contain some carbohydrates, fats and all of that, right? There is some ghee, some dal which will contain proteins and some roti probably that will have starch, carbohydrates. So you've consumed that. In your digestive system, it'll get broken down into its constitutive particles. So let's consider glucose for example, because that is the substrate that the cells will mostly prefer. Now this glucose that's broken down inside your digestive system will enter your blood capillaries and the blood is getting transported throughout your body to every one of your living cells. Okay. So this glucose has 6 molecules, 1, 2, 3, 4, 5, 6 with these carbon-carbon bonds. This is not like an exact structure of glucose. For understanding, I'm just writing it. Okay, so there are 6 carbons. In between these carbons, bonds are there, chemical bonds are there. Whenever this gets broken down, energy will be released. Okay. Now, how is this energy going to be stored in our cells? Right now, glucose is getting broken down. Some amount of energy is getting released. But the cell may not require that energy at that moment. Okay. So, what basically happens here is, when glucose or any other substrate for that matter, which has carbon-carbon bonds, is getting broken down, the energy that is released is trapped in another molecule. If you don't trap it in another molecule, it gets wasted as heat. It gets lost as heat. You know, when anything is getting broken down, energy will be released, right? So even here, energy is released and we are trapping that energy in another molecule called ATP. You may have heard about ATP. It is called adenosine triphosphate. So that is also called the energy currency of the cell. I'll tell you why it's called the energy currency. So this is a glucose molecule. When it gets broken down, carbon dioxide will get liberated and energy is released. This energy is stored or trapped in molecules called ATP. ATP is adenosine triphosphate. I'm sure you've all studied about adenosine in the chapter biomolecules. Adenosine is a nucleoside. Generally, the nucleotides in our DNA and RNA will contain one phosphate group attached to the 5' carbon. But this adenosine triphosphate also is a nucleotide, or you can call it a nucleoside triphosphate. But here at the 5' end, 3 phosphate groups will be attached. Okay. So this is the structure of adenosine triphosphate or ATP. In the center, you will have a sugar which could be RNA or DNA. To one end, nitrogenous base adenine will be attached. To the other end, 1, 2, 3, 3 phosphates will be attached. So at this time, when glucose is getting broken down, energy will be released, right? That energy is utilized to synthesize this molecule called ATP. How exactly does this get synthesized? In our cells, you will have adenosine diphosphate. Di means 2. This is the same adenine, this is the same pentose sugar. It will have 1 to 2 phosphates. And here you have an inorganic phosphate. Utilizing that energy, this phosphate gets attached to this molecule forming adenosine triphosphate. Here bond is breaking, energy is released. That energy is utilized to create a bond here and synthesize ATP. Now, whenever the cell has to perform a metabolic reaction which requires ATP, which requires energy, then what happens is, at that moment, to provide energy for that particular chemical reaction, this ATP will break down. The bond that we have here between the second phosphate and the third phosphate is a high energy bond. When this breaks, considerable amount of energy is released, and that is what the cells use to perform their metabolic activity. So the glucose that is getting broken down, energy that is released will not be utilized directly for metabolic activity. Whatever energy is released that energy is trapped inside this molecule. The energy that is released here is utilized here to synthesize this molecule. This bond here between the second and third phosphate is a high energy bond. Whenever at whatever time the cell requires energy, this molecule will break. ATP will become again ADP and inorganic phosphate. When this bond here breaks, energy is released. That energy will be utilized by the cell to perform metabolic activities. Alright, I hope that is clear. Alright, here is an interesting question. The question is, do plants breathe? We breathe, like most animals breathe, higher animals breathe. We've learnt about, breathing as a physical process, even in breathing and exchange of gases, human physiology chapter, where we have mechanisms to draw air into our lungs and exhale, give out air and all of that. That is the whole process of breathing. And different animals have different respiratory organs and structures that assist in this breathing. Why do we breathe? Why do I need this respiratory structure? Why do I need this respiratory system? Why should I have nostrils and a trachea and lungs and alveoli and all of that? Because every part of my body has cells that require oxygen, right? The cells in my toe also require oxygen. The cells in my brain require oxygen. The cells in my kidney require oxygen. So all of these organs require oxygen. And they themselves are not directly in contact with the atmosphere. There's no way there is any contact between the cells inside my kidney, the cells inside my brain to the atmosphere, right? The reason we don't have that kind of access is because around us there are so many potentially disease causing microbes present. If we also had stomata like plants along with air, the disease causing microbes also could enter us, right? That's why we do not have that much access to the external environment. Every part of our body does not have that kind of structure which can allow gaseous exchange. That is why the lungs do the job for all of us. You have nostrils through which you take in air. In that air, oxygen will be present. Because of a difference in the partial pressure of oxygen, whatever oxygen is there in your lungs, that will get transported to your blood vessel. And the blood will carry that oxygen to every part of our body, supplied to all of our living cells. In the living cells, when oxygen is used to break these food molecules, carbon dioxide is released as a byproduct. The blood will pick up that carbon dioxide and bring it back to the lungs and we exhale it, right? This is what happens. The reason why we have complex respiratory structures like lungs, trachea, bronchioles and all of that is because every part of our body cannot take care of its own respiratory needs. Every part requires oxygen, but every part doesn't have access to oxygen. It doesn't, right? Therefore, our respiratory system does, with the help of circulatory system, of course, does the job of supplying oxygen to all of our cells and also taking back carbon dioxide from the cells and exhaling it. But plants can survive even in the absence of these respiratory structures. They do not have lungs, they do not have a respiratory system. Why is it that, or how is it that plants can survive even in the absence of these respiratory structures is a question that we should ask ourselves. There are three important reasons why plants can survive even in the absence of respiratory structures. First of all, do plants breathe? That is the question that we started off with, right? We know generally we say animals, we take in oxygen, give out carbon dioxide. Plants take in carbon dioxide, give out oxygen. That is true, all right? Plants do take in carbon dioxide and give out oxygen, but that is in photosynthesis. Every living organism undergoes respiration as well. For the process of respiration, plants also require oxygen and plants will also release carbon dioxide. So, when respiration is happening, plants also need oxygen and release carbon dioxide. Alright, so in us, respiration happens, photosynthesis does not happen. In plants, photosynthesis also happens, respiration also happens. For photosynthesis, carbon dioxide from the atmosphere is taken in, oxygen is released back to the atmosphere. For respiration, oxygen is utilized, carbon dioxide is released, the opposite happens, alright. Whether or not plants breathe, like the physical process of breathing, is difficult to answer. There are no structures that will help in breathing, but of course, even in plants, exchange of gases will take place for photosynthesis as well as respiration. What are the 3 reasons why plants can survive even in the absence of respiratory organs? We'll understand that first. The first important reason is, unlike us, where every part cannot take care of its own gaseous exchange needs, it depends on our lungs and circulatory system. In plants, it's not like that. In plants, every part can take care of its respiratory needs, take care of its gaseous exchange needs. What are the different organs that you see in a plant? You have root, you have stem, you have leaves, and of course the reproductive structures, right? The leaves will exchange gases for itself. Whatever is required for respiration, leaf will take care of its own needs. How can leaf exchange gases? We've learnt in the chapter Anatomy of Plants that leaves have tiny pores, openings called stomata present in the epidermal layer, and through that exchange of gases can happen both for photosynthesis and respiration. So leaf will take care of itself. What about the stem? If the stem is young and green, it will have some stomata present on its surface through which gaseous exchange can happen. What if it's like a big stem, big fat trunk? In woody stems, you have openings, lens-shaped openings that are called lenticels present, and through these lenticels, exchange of gases will take place. Alright? First important point is, every part in a plant will take care of its own gas exchange needs. Secondly, plants do not have too much demand for oxygen when it comes to respiration. They don't have to depend on atmospheric oxygen much, because in plants photosynthesis is also taking place. The end product of photosynthesis is oxygen. That oxygen is getting released inside the cell. That oxygen itself can be used for the process of cellular respiration. So they don't have to entirely depend on the atmospheric oxygen when it comes to respiration. Of course, gaseous exchange happens, but that happens mostly for photosynthesis and it's not much for respiration. Alright, because oxygen is directly available for them. As an end product of photosynthesis, they will utilize the same oxygen. They don't require much oxygen from the atmosphere. So the demand for oxygen for respiration in plants is not as high as it is in animals. Alright, also the rate at which plants respire is much lower compared to the rates at which we respire. Alright, that's the reason they can survive even in the absence of respiratory organs. What is the third most important point? The distance that gases must travel is larger in us. Lungs have to carry oxygen from the lungs. Blood has to carry oxygen from our lungs to our brain, to our feet, to our fingers, everywhere. So it has to travel a long distance. That is why we have all of these organs and organ systems that help in the process. But in plants, that is not the case. You might argue, I understand that if it's a leaf, it is very thin. It has a few cell layers in thickness. Maybe over there, transfer or movement of gases, the distance that the gas has to travel is less. What about thick trunks? What about big plants that have like very woody thick stems and trunks? What is the case then? If you remember Anatomy of Flowering Plants, I've told you how in big trees or even in stems, there will be cortex present, and in the cortex, the cells are loosely arranged like this. There will be a lot of air spaces. If you think about thick woody stems, whatever is there in the center generally is dead tissue. We call it the heartwood. It will have a lot of dead cells that don't even need oxygen because they are dead, right? They are there for mechanical support. They are there for protection from, infestation and disease. But they are not living, so they don't need oxygen. Oxygen is required only by the cells that are present in the surrounding layers where living cells like parenchyma, collenchyma may be present. So only those cells require oxygen. And in the bark, we will have structures called lenticels that will have access to external atmosphere. So oxygen is taken in from those parts. And because there is so much air spaces, it's easy for this oxygen to move through these air spaces and get delivered to every one of the living cells. So since the distance that the oxygen has to travel or the gases have to travel is not very large, It is manageable even in the absence of respiratory organs. Okay, firstly, every part takes care of its own gas exchange needs. Secondly, there is not much great demand for oxygen in plants. Mostly gaseous exchange happens for photosynthesis, and the rate of respiration is also not very high in plants compared to animals. Thirdly, the distance that the oxygen has to travel in animals is much larger, which requires specialized systems, and that is not the case in plants, because even though we are talking about huge trunks, the center part will usually be dead. The surrounding bark layer will have dead cells. In between whatever living cells are there, they will be loosely arranged. So what oxygen can easily diffuse through these air spaces and be obtained by all the living cells in a plant. Therefore, they can survive even in the absence of respiratory organs. Okay, now let's talk about respiration of glucose. In this chapter, focusing on how energy is obtained when glucose gets broken down. We're focusing on how it is for glucose, because glucose is the most preferred substrate for any cell. Okay, so when glucose combustion happens outside a living body, whenever glucose is burnt or combustion happens, in that process carbon dioxide will get released and energy is liberated in the form of heat, right? In our cells, what we do is, we do not burn glucose or oxidize glucose in one step. In our cells, glucose get oxidized in many small steps. And some of these steps are just small enough and liberate just enough energy so that one ATP molecule is synthesized. For ADP to combine with inorganic phosphate and synthesize ATP, energy is required. How much energy is required for that? There is one number, one value. Let's consider it X. Okay. When glucose is getting combusted, burnt, oxidized in one step, then the energy will be lost as heat. We do not want that. We've eaten food, we need energy later. We want to conserve whatever energy is there in glucose in another molecule called ATP. So instead of burning glucose in one step and losing out on all the energy in the form of heat, what we do is, we break glucose down in multiple steps that are very, very small. Like many steps, it will get oxidized, and some of these steps will produce that X amount of energy that is required for ATP synthesis. So that much, just that much energy is liberated so that one molecule of ATP can get synthesized. All right. So by breaking down glucose, oxidizing glucose in many small steps, we synthesize or utilize the energy in the synthesis of ATP. Now glucose will have some amount of energy. All of the energy present in glucose cannot be trapped as ATP. Only a percentage of that can be trapped as ATP. Some amount will be lost as heat. Okay, even inside our cells, 100% of the energy present in glucose cannot be stored as ATP. Okay, some amount, how much ever possible, our cells will try and utilize and store that and trap that energy in the form of ATP molecules, which can be used wherever required. Okay, so the strategy is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key here is to oxidize glucose not in one step but in several small steps, enabling some steps, only few steps of those steps, to be just enough so that energy that is released can be coupled to the synthesis of ATP. Alright, now every time I have been saying that glucose gets oxidized and energy is released, glucose gets oxidized, and I'm writing also oxygen being used. But we know that in nature, there are many organisms that have the ability to survive without oxygen. Some may be facultative anaerobes where, like our mycoplasma, about which we've learned in the chapter biological classification. Mycoplasma can survive in oxygen, can survive in the presence of oxygen, can survive in the absence of oxygen as well. There are some organisms that are strict anaerobes, obligate anaerobes, which will die when oxygen is there. They require anaerobic conditions to survive. And we all are aerobic organisms, we need oxygen to survive. So this equation kind of makes sense, utilizing oxygen to get energy kind of makes sense in aerobic organisms, right? Aerobic or anaerobic, we need energy to survive. We need ATP to survive. I'm saying ATP is getting produced when oxygen is used for breaking down glucose. But I'm also saying that some organisms survive only in the absence of oxygen. If those organisms cannot stand oxygen, if those organisms require ATP to survive and they can't stand oxygen, then how do they get energy? I'm saying that for respiration, oxygen is required. I'm saying all organisms require ATP for survival. There is one organism that needs ATP, it can't stand oxygen. How is respiration going to happen in that case? In respiration, there is one step called glycolysis, which will happen in the absence of oxygen. That process does not require oxygen. And in that process, glucose is not fully oxidized, it will only get partially oxidized and some amount of ATP can be formed. So those organisms which are strict anaerobes or even facultative anaerobes, they can obtain their energy by that one process which doesn't itself consume oxygen. All right? Yeah. All living organisms retain the enzymatic machinery. You know how every reaction that takes place in our cells is catalyzed by enzymes. For that partial oxidation without the presence of oxygen, whatever enzymes are required, that is present in all of our cells. So when organisms are anaerobic, they can produce ATP by that one process. Okay, so to partially oxidize glucose without the help of oxygen. And that process is what we call glycolysis. Lysis means breakdown. Glyco means sugar. Here we are talking about the breakdown of glucose, right? So glycolysis is a process where glucose will partially get broken down. Glucose will not break down to form 6 molecules of carbon dioxide. Full oxidation will not happen. Glucose will partially get oxidized in the absence of oxygen. It's not like glycolysis will not happen if oxygen is there. What I'm trying to say is glycolysis can happen even if oxygen is there, but glycolysis itself does not require oxygen. Oxygen is not involved in this process, okay? So it can happen in both aerobic and anaerobic organisms. Glycolysis is a 10-step reaction where one 6-carbon molecule called glucose gets broken down into 2 molecules of a 3-carbon compound called pyruvic acid. Alright, glucose, as we know, contains 6 carbons. This glucose in the process of glycolysis will be partially oxidized and it will form 2 molecules of pyruvic acid which contains 3 carbons. 2 3 times is 6. So this 6-carbon compound gets broken down into 2 3-carbon compounds, and when that partial oxidation or breakdown happens, some amount of energy can be trapped in the form of ATP. This is the first step of cellular respiration. This happens in anaerobic organisms as well as aerobic organisms, and this occurs in the cytoplasm. Okay, so over here, remember, glucose is only partially oxidized. Now, this glycolytic pathway is also called EMP pathway. Where did we get this EMP from? There are 3 scientists, 3 researchers who elucidated the whole 10-step reaction, what exactly is happening in each reaction. And their names are Emden, Meyerhoff, Parnas. E for Emden, M for Meyerhoff, P for Parnas. So to, give regard to these scientists for your contribution, we also call glycolysis as EMP pathway. Remember, this is a process that occurs in aerobic organisms as well as anaerobic organisms. In aerobic organisms, this process will produce some amount of ATP, but after this there are other processes which utilize oxygen and produce more ATP. But in anaerobic organisms, this is the only process that will help in generation of energy. And this glycolysis process does not need oxygen. Now, there are some doubts that students ask me generally. I am saying that it does not need oxygen. I am also saying glucose is getting partially oxidized. If oxygen is not there, how is it getting oxidized? Now I want you to understand that getting oxidized does not have anything to do with oxygen. Okay, what is oxidation that you've learned in chemistry? Oxidation is losing of electrons, right? We are talking about that oxidation. Electrons are getting lost. Oxidation doesn't necessarily require oxygen. If electrons are getting lost, then we call it oxidation. Here, that oxidation is happening partially. Complete oxidation, if it happens, all the 6 molecules of carbon dioxide will be released. Here, partial oxidation is taking place. For oxidation, oxygen is not required. All right. Now, let's move on and learn about how exactly glycolysis takes place. When photosynthesis takes place in plants, glucose is produced, right? Glucose is a product of photosynthesis. Now, if this glucose has to be transported within the plant, I told you how some parts of the plant cannot synthesize food. Leaves can, but the roots can't. Some cells inside the stem can't, right? So, for those parts, for those cells, glucose that is produced by the leaves have to be transported, translocated. And that happens via phloem. And when glucose is getting transported, it is not transported as glucose. Instead, it is transported as sucrose. All right. Now, sucrose is a disaccharide. Sucrose will contain glucose and fructose. One glucose, one fructose coming together will give sucrose. We have glucose from photosynthesis. How did we get fructose? Glucose and fructose are isomers. Glucose and fructose have the same chemical formula but different structures. Some glucose molecules will isomerize to form fructose. So one glucose, one fructose will combine to form sucrose, and sucrose is what the plants use to transport food. Alright, it is transported as sucrose across different parts of the plant. Now, if photosynthesis has happened and there is a lot of glucose synthesized, you do not know if tomorrow sufficient water will be available for photosynthesis, if the sunlight will be conducive for photosynthesis. All of the excess glucose that you've produced in the cell, you don't require it right now, so you want to store it for future. One day, if there is not sufficient sunlight for photosynthesis, you can utilize that. If there's not sufficient water in the soil, you can utilize that. So as a reserve food, you want to store some glucose. Glucose is not stored as it is. It forms a polymer called starch. So whenever glucose has to be stored, it is stored as starch. Whenever it has to be translocated, it will be in the form of sucrose, which is a disaccharide containing glucose and fructose. All right. When glucose enters the pathway, I'll tell you what happens. This sucrose, which is a disaccharide, first it will get broken down. This breakdown will help in the formation of glucose and sucrose. Glucose and sucrose, both of them are 6 carbon containing molecules. And for sucrose to break down and form glucose and fructose, you need an enzyme called invertase. So in the first step, the sucrose, which is a disaccharide, will get broken down into glucose and fructose, which are both 6 carbon molecules, by utilizing the enzyme, or which is catalyzed by the enzyme called invertase. Now this glucose and fructose will readily enter the glycolytic pathway. We are going to start the glycolytic pathway with glucose. Alright. In glycolysis, I told you have a total of 10 steps, right? We have to learn about what exactly happens in each of those steps. Let's start off with the first step. In the first step, glucose which contains 6 carbons it does not contain any phosphate, 0 phosphate. Okay. This glucose 6-phosphate will get converted, or it will get phosphorylated to form glucose 6-phosphate. Now this glucose 6-phosphate has 6 carbons but we are calling it glucose 6-phosphate. So to the 6th carbon of glucose, a phosphate group is attached. So this now has 1 phosphate. Where did this 1 phosphate come from? Where did phosphate come from for phosphorylation of glucose? Glucose has 6 carbons. To the 6th carbon, 1 phosphate group is attached. Where did that phosphate come from? This phosphate is because of ATP. A molecule of ATP has 3 phosphates. I've told you that earlier. It gets broken down, releasing 1 phosphate. It becomes ADP and inorganic phosphate. This inorganic phosphate gets added to glucose, and that phosphate gets added to the 6th carbon. So from glucose, you get glucose 6-phosphate. This reaction is catalyzed by an enzyme called hexokinase. Actually, every step has an enzyme that catalyzes it, but in your syllabus, you only have to learn about the name of this one enzyme, which is hexokinase. Now, this glucose 6-phosphate will get isomerized and form fructose 6-phosphate. Fructose 6-phosphate also, since it's a isomer of glucose, will contain 6 carbon, and it will also have a phosphate present in the 6th carbon. So it's called fructose 6-phosphate. So I'm going to write 1 phosphate here. What's going to happen next? Another phosphorylation will take place. So to the 1st carbon of fructose also another phosphate group will get attached. So fructose 6-phosphate will get phosphorylated and form fructose 1,6 bisphosphate. Bisphosphate means phosphate groups are attached to 2 different carbons. One is the 1st carbon, the second is the 6th carbon. So here fructose, there is no change in the number of carbons. This will have 6 carbons, but now it has 2 phosphate groups, one attached to the 1st carbon, the other one attached to the 6th carbon. There was only one in the 6th carbon. If a new phosphate is attached to the 1st carbon, where did that phosphate come from? Another molecule of ATP will get broken down here to ADP and inorganic phosphate, and that inorganic phosphate is attached to the 1st carbon of fructose, making it fructose 1,6 bisphosphate. I've been telling you that whenever glucose gets broken down, energy is released in the form of ATP. We try to trap that energy in the form of ATP, but right now if you look at it, we're not synthesizing ATP. We are utilizing ATP, right? ATP is getting used, ATP is not forming. In the first 5 steps, which is called the preparatory phase, ATP will get utilized. In the next 5 steps, ATP will form. Alright, it's like these schemes that you have in banks. You deposit 1 lakh, come back to us after 2 years, we'll give you 2 lakhs. It gets doubled, right? Like that, even over here in the first phase, how many ever ATP is used, in the next 5 steps, twice the amount of ATP will get synthesized. Alright, so these are the first 3 steps. In the next step, this fructose will split to form 2 molecules. Phosphoglyceraldehyde which is abbreviated as PGAL, and dihydroxyacetone phosphate which is abbreviated as DHAP. Now each of these contains 3 carbons. We had 6 carbon molecule and 2 phosphates. So each of these will have 3 carbon present and each one will have 1 phosphate. Similar to how glucose and fructose are isomers, DHAP and PGAL, they are isomers. So in the 5th step, they will get isomerized. Whatever dihydroxyacetone phosphate is formed, that will also get converted to phosphoglyceraldehyde. So from the next step onwards, which is the 6th step, so far we have 1, 2, 3, 4 is splitting, 5th is isomerization. So 5 steps are over. In 5 steps, 2 ATPs are utilized. At the end of the 5th step, we have 2 molecules of phosphoglyceraldehyde. From the 6th step onwards, we are going to start with 2 molecules of phosphoglyceraldehyde. Okay. From the next step onwards, we will have 2 molecules. Now, I have 2 molecules of phosphoglyceraldehyde. Okay. Now, this phosphoglyceraldehyde has 3 carbons and 1 phosphate attached to the 3rd carbon. I've been saying that glycolysis is partial oxidation. This is the step in which oxidation actually takes place. You know, when an aldehyde gets converted to an acid, aldehyde is getting oxidized to form an acid, right? So phosphoglyceraldehyde in this step will get oxidized and forms 2 molecules of 1,3 bis phosphoglyceric acid. Now this step is important because of 3 reasons. This is important. Firstly, because this is the exact step where oxidation is happening. First most important thing, I've been saying that it will get partially oxidized. So oxidation is basically happening in this step. Alright. Secondly, as phosphoglyceraldehyde or glyceraldehyde 3-phosphate, you can call it either, as this is getting oxidized to form 1,3 bis phosphoglyceric acid, another thing that you can notice is, this phosphoglyceraldehyde will give 2 hydrogen in the form of redox equivalents to a molecule of NAD+. So, of the 2 redox equivalents, one will be a proton, the other will be a hydride ion. So, this is picked up by a molecule of NAD+, not NADP. NADP is in photosynthesis. Here it's NAD+. It's the same thing, nicotinamide adenine dinucleotide. We've learnt about it in photosynthesis, right? Same thing. So, but there is no phosphate here. So, this NAD+ will take up this proton and hydride ion that this molecule has given, and it will get converted to form NADH because it picked up this hydride ion, and in addition to that it will have a proton. So phosphoglyceraldehyde will donate 2 redox equivalents to NAD+ molecule. Of the 2 redox equivalents, one is a proton, one is a hydride ion. So the proton, the hydride ion binds to NAD+. Since it already has 1 less electron, it accepts H- and becomes NADH, and you will have this additional H+. Okay. So as aldehyde is getting oxidized to acid, NAD+ will get reduced to NADH. Now what is this NADH? NADH also is an energy rich molecule. It is equivalent to 3 ATP molecules. ATP is like currency, money, cash that you can use right away. NADH is like a check. if NADH has to be used, it has to be taken to the bank. You have to get it encashed and then you can utilize it. So this NADH also has to go to a bank where it gets converted to ATP. Each molecule of NADH is worth roughly 3 ATP. So it has to go to the bank and that bank is present in the mitochondria. Okay, so more about it we'll learn as we move on. For now, just remember this NADH also is a high energy molecule that will give us more ATPs. So here, for each molecule, one NADH is produced. Now that we have two molecules like this, 2 NADH will be produced. Okay, so 2 NAD plus will combine to form 2 NADH and H plus. Alright, another important thing that I want you to notice is, here we had phosphoglyceric acid that had 3 carbons and 1 phosphate. Here 1,3 bisphosphoglyceric acid has 3 carbons but it has 2 phosphate. Where did this additional phosphate come from? Unlike what happened previously where ATP used to get utilized for addition of phosphate or phosphorylation, over here phosphoric acid 3PO4 that will be utilized for donating phosphate group to this molecule so that it will have 3 phosphates. Okay, so this is the step in which oxidation is happening. This is the step in which NAD plus is getting reduced to NADH and H plus, and this is the step where for phosphorylation ATP is not getting utilized, phosphoric acid is getting utilized. These 3 things are important, you should keep that in mind. Alright, now this 1,3 bisphosphoglyceric acid will next get converted to 2 molecules of 3-phosphoglyceric acid. In 3-phosphoglyceric acid, here you had 1 phosphate attached to the first carbon and third carbon. Here you have 1 phosphate attached only to the third carbon. Here you had 2. Here you had only 1. The number of carbons, there is no change, but there is a change in the number of phosphates, right? What happened to that additional phosphate? That is utilized right now to form ATP molecule. From ADP, ATP will get formed. Here ATP got utilized. Look at how the arrows are. And here ATP is forming. Where did the phosphate come for the formation of ATP? It came from here. Okay, so ATP synthesis happens. Per molecule 1 ATP. Here we have 2 molecules, so we have 2 ATPs that will get synthesized in this step. In the next step, 3-phosphoglyceric acid will get converted to 2 molecules of 2-phosphoglyceric acid. This will have 3 carbons. It will have 1 phosphate. The only difference is these both are positional isomers. In 3-phosphoglyceric acid, phosphate group was attached to the third carbon. In 2-phosphoglyceric acid, that will be moved to the second carbon. So third carbon, 3-phosphoglyceric acid, second carbon, 2-phosphoglyceric acid. Now in the next step, 2-phosphoglyceric acid will get converted to a molecule called phosphoenol pyruvate. 2 molecules of phosphoenol pyruvate. This phosphoenol pyruvate also contains 3 carbons. It will also contain 1 phosphate. So there's no change in the number of carbons or the number of phosphate. But over here, in this step, you can see that a molecule of water will get released. Next, we have the last step. What is the end product that I mentioned? Pyruvic acid is going to form. So in the last step, 2 molecules of pyruvic acid are going to form. Pyruvic acid has 3 carbons, but it does not have any phosphate. What happened to the phosphate that this had? It will be utilized in the formation of a molecule of ATP from ADP. Since here you have 2 molecules, we will form 2 ATP. I want you to notice how one 6-carbon compound is now broken to two 3-carbon compounds, and this has happened over a series of 10 steps. Some of these steps, which is here and here, the breakdown has been small enough that ATP could be synthesized. It is getting oxidized at the 6th step. In the first 5 steps, 2 ATP molecules are utilized. In the next 5 steps, we got 2 plus 2, 4 ATP synthesized. So totally in glycolysis, 4 ATP synthesized, 2 are utilized. So how much remains? What is the net gain? Net gain will be 2 ATP. Did we get anything else from this process? We also got an energy rich molecule called NADH. Not one but 2 molecules of NADH. Alright, so that's about glycolysis. You should remember exactly in which step what is happening. So I've made like a table for you to remember when ATP is utilized, when ATP gets synthesized, when NADH is produced, when water is released. For all of that, you have information in this table right here. So how much ATP did we gain? We totally gained 4, of which 2 was utilized. Therefore, we are left with only 2. How much NADH did we get? We got 2 NADH in this process. I want you to keep this in mind. We will need this later. Alright. Now, what happens to this pyruvic acid? Now, this pyruvic acid can either undergo fermentation anaerobically or undergo aerobic respiration. If it undergoes fermentation without utilizing oxygen, we call it anaerobic respiration, and two things can happen. Either this pyruvic acid can get decarboxylated and form ethyl alcohol or ethanol, in which case we call it alcoholic fermentation because alcohol is formed as the end product, or it can get converted to lactic acid, which is a 3-carbon compound. In this case, decarboxylation will not take place. And this process is called lactic acid fermentation because ultimately lactic acid will be the end product. Okay, first let's learn about alcoholic fermentation. You can see alcoholic fermentation happening in organisms like yeast. You know, yeast is utilized for the production of alcoholic beverages, right? So, Saccharomyces cerevisiae. Yeast undergoes alcoholic fermentation and produces ethyl alcohol as the end product. That is how we get or obtain alcoholic beverages. So over here, pyruvic acid that we got at the end of glycolysis will first undergo decarboxylation, where a carbon dioxide gets removed, one carbon is removed, and it forms acetaldehyde which contains 2 carbons. When pyruvic acid is getting converted to acetaldehyde, the enzyme that catalyzes this reaction is called pyruvate decarboxylase. Understand that pyruvic acid is getting decarboxylated, so the enzyme is called pyruvate decarboxylase. In the next step, this acetaldehyde will get, reduced to ethanol. Aldehyde to alcohol is reduction, right? So aldehyde is getting reduced to alcohol. Both of them contain 2 carbons. And the enzyme that catalyzes this is called alcohol dehydrogenase. As aldehyde gets reduced to ethanol, NADH will get reoxidized to NAD+. Whatever NADH was was synthesized during glycolysis, step number 6, that will get reoxidized to NAD+ in alcoholic fermentation. So in organisms that undergo alcoholic fermentation, the only amount of energy that they will obtain is 2 ATP molecules that they got from glycolysis. Even the NADH that was produced gets reoxidized here, so even that is not of much help, right? That's about alcoholic fermentation. Now talking about lactic acid fermentation, this occurs in certain bacteria like lactic acid bacteria, which is involved in the formation of curd, and also in our muscle cells when we don't breathe enough or we don't breathe properly. Our muscles are not getting sufficient oxygen during strenuous exercise. At that time, lactic acid fermentation can happen in our muscle cells as well. So what basically happens here is, there is no decarboxylation. 3-carbon molecule pyruvic acid gets converted to 3-carbon lactic acid, and in this process the enzyme that is catalyzing is called lactate dehydrogenase. Even during lactic acid fermentation, you can see how The NADH that was synthesized during glycolysis will get reoxidized to NAD plus NADH plus H plus will get reoxidized to NAD plus. So this is what happens during lactic acid fermentation. So in lactic acid fermentation or alcoholic fermentation, both fermentation types or anaerobic respiration, the amount of energy that we get from glucose is very, very little. In those organisms, the next steps of respiration will not take place. Whatever energy they obtain from glycolysis, that is all they will get. And that energy is also, whatever energy is there in glucose, less than 7% of energy is released, alright, during this partial oxidation, during glycolysis, less than 7% of energy is released. Even among that energy that is released, all of it will not be trapped in the form of ATP, so very little energy is obtained, only 2 ATP molecules is obtained. In contrast, when oxygen is utilized in aerobic respiration, which we will see further, you can see how lot many ATP molecules will be synthesized, alright. So this fermentation process has two drawbacks. Firstly, like I already mentioned, energy released is not very high. Less than 7% of energy that is there in glucose is released, and whatever is released completely is not trapped in the ATP molecule. So that is a disadvantage. Secondly, this process can be hazardous because the end product is either an alcohol or an acid, which in high quantities can be detrimental for the functioning of the cell. Okay. Now, I told you how yeasts are organisms that are involved in the commercial production of alcoholic beverages. It undergoes alcoholic fermentation in the absence of oxygen. So whenever yeast is growing in a medium anaerobically, it is undergoing alcoholic fermentation. It is producing alcohol as a product, and that alcohol will be present in its surroundings. So when in the medium it is growing, the concentration of the alcohol, if it comes around 13%, that is all yeast can handle. Beyond that, yeast will not be able to handle. That will become toxic for the yeast itself, and the yeast will start dying. Okay. So yeast poison themselves to death when the concentration of alcohol reaches about 13%. But if you notice, there are certain alcoholic beverages in which alcoholic content is greater than 13%, like whiskey, brandy, rum and all of those. The alcohol content is greater than 13%. If yeast itself is dying at a concentration around 13%, how are they able to obtain alcoholic beverages that have greater than 13% alcoholic content? That is by a process called fractional distillation. You don't have to worry about it right now. You will learn more about it in another chapter in 12th grade called Microbes in Human Welfare. For now, just remember that it's called fractional distillation. You don't have to know more about it. Alright, that's about fermentation. Now let's talk about what happens to pyruvate in aerobic respiration. Remember, the whole process of glycolysis took place in cytoplasm. Aerobic respiration is going to take place in the mitochondria. So whatever pyruvic acid is produced in the cytoplasm will be transported, will be taken into the mitochondrial matrix for the next step. The next step is called the link reaction or oxidative decarboxylation. We know that pyruvic acid contains 3 carbons. In this link reaction or oxidative decarboxylation that takes place in the matrix of the mitochondrion, you can see how simultaneously oxidation and decarboxylation will take place. Over here, pyruvic acid, which contains 3 carbons, will get decarboxylated. Carbon dioxide will be removed. 1 carbon is removed and it forms acetyl-CoA, which is a 2-carbon compound. In order to form this acetyl-CoA, pyruvic acid will react with a coenzyme called coenzyme A, which is represented as CoA. So all of this is happening in the mitochondrial matrix. Pyruvic acid reacts with coenzyme A and it will get oxidized to form acetyl-CoA. Simultaneously, decarboxylation is also happening. And in this process, while pyruvic acid is getting oxidized to acetyl coenzyme A, NAD plus will get reduced to form NADH plus H plus. Similar to how NAD plus was reduced during the 6th step of glycolysis, over here also NADH plus H plus will form. NAD plus will get reduced to give NADH and H plus. Okay, I repeat. This is called the link reaction or oxidative decarboxylation. This is what happens only in aerobic organisms. This is happening during aerobic respiration, and this is happening in the matrix of the mitochondrion. During oxidative decarboxylation, oxidation also happens, decarboxylation also happens. Pyruvic acid reacts with coenzyme A, undergoes decarboxylation and oxidation to form acetyl coenzyme A, which is a 2-carbon compound, and carbon dioxide molecule is released. While pyruvic acid is getting oxidized to acetyl coenzyme A, NAD plus will get reduced to form NADH and H plus. Alright, so this is catalyzed by an enzyme called pyruvate dehydrogenase, and this requires the presence of magnesium ions. Now, whatever acetyl coenzyme A has formed by the process of oxidative decarboxylation or link reaction will enter a cyclic pathway within the mitochondrial matrix, which is called the Krebs cycle. Why it is called Krebs cycle is because it was elucidated by a person called Hans Krebs. Now, this Krebs cycle has alternate names as well. Krebs cycle is also called citric acid cycle, because the first product that we obtain in Krebs cycle is citric acid. So you call it the citric acid cycle as well. Okay, so this is also called citric acid cycle, and it is also called TCA cycle. TCA cycle is short for tricarboxylic acid cycle. Citric acid and some intermediates in this pathway contain 3 carboxylic acid groups. That's why it's also called tricarboxylic acid cycle. So these are the alternate names for Krebs cycle. Krebs cycle, TCA cycle, tricarboxylic acid cycle, citric acid cycle, all of these mean the same thing. Okay. So what basically happens during Krebs cycle, I will explain to you right now. Pay attention. We got acetyl-CoA from the link reaction. Now this acetyl-CoA contains 2 carbons. This acetyl-CoA will undergo condensation with a 4-carbon organic acid called oxaloacetic acid, abbreviated as OAA. Remember, we've learnt about it in photosynthesis as well. In C4 plants, the first stable product formed is oxaloacetic acid. It has 4 carbons. We know that, right? So this acetyl-CoA containing 2 carbons will react with oxaloacetic acid that contains 4 carbons. 2 carbon plus 4 carbon will give us a 6 carbon acid, organic acid called citric acid. Now for the formation of citric acid, you require an enzyme called citrate synthase. This enzyme catalyzes the first step in Krebs cycle. What is happening? Acetyl-CoA, which we got from previous step link reaction, undergoes condensation reaction with oxaloacetic acid, which contains 4 carbons, and it forms citric acid, which contains 6 carbons. Now, during this process, this process is catalyzed by an enzyme called citrate synthase, and during this process, coenzyme A molecule will get released. Okay, acetyl coenzyme A had coenzyme A that will get released. Acetyl-CoA, oxaloacetic acid combined to form citric acid. Now, this citric acid has 6 carbons. In the second step, the citric acid will get isomerized into another molecule, an isomer obviously, which also contains 6 carbons, and that is called isocitrate. So in the second step, there is no change in the number of carbons. First step, 2 plus 4 gave us a 6 carbon compound. Second step, isomerization is happening. Citric acid is getting converted to isocitric acid or isocitrate. That is our second step. Now in the third step or the next step, this isocitric acid, which contains 6 carbons, will undergo decarboxylation, which means 1 carboxyl group is removed as carbon dioxide. And if 1 carbon is gone, what remains? 5 carbons remain. So this isocitric acid will undergo decarboxylation to form a 5 carbon compound called alpha-ketoglutaric acid. Okay, so in our next step, this 6 carbon isocitric acid is undergoing decarboxylation. 1 carbon is removed in the form of carbon dioxide, and it forms a 5 carbon compound called alpha-ketoglutaric acid. I want you to notice over here how, similar to in the link reaction, oxidation of isocitrate is happening to alpha ketoglutaric acid. Oxidation is happening simultaneously, decarboxylation is also happening. So you can call this reaction also as oxidative decarboxylation. So similar to how in link reaction we saw that simultaneously there was reduction of NAD plus to NADH and H plus, it is happening here as well. So in the third step, isocitrate or isocitric acid is undergoing oxidation and decarboxylation simultaneously to form a 5-carbon alpha ketoglutaric acid, and in this process carbon dioxide is removed. As isocitric acid gets oxidized simultaneously, NAD plus will get reduced to form NADH and H plus. Alright, now let's take a look at our next step. In the next step, 5-carbon alpha ketoglutaric acid will again undergo decarboxylation. So this 5-carbon alpha ketoglutaric acid will lose another carbon in the form of carbon dioxide molecule and form a 4-carbon compound called succinyl coenzyme A. Alright, so even during this process, a molecule of NADH plus H plus will get formed. NAD plus will get reduced to form NADH and H plus. We saw decarboxylation happening in link reaction. We saw decarboxylation happening from isocitrate to alpha ketoglutarate right here. From alpha ketoglutarate to succinyl coenzyme A, we saw decarboxylation happening. I told you, in aerobic respiration, complete oxidation of glucose will happen. What do I mean when I say complete oxidation of glucose will happen? I mean that glucose will be completely broken down to give 6 molecules of carbon dioxide. Right now we have already obtained 6 molecules of carbon dioxide. How? See, here you have 1 carbon dioxide, right here. Here you have the 2nd carbon dioxide. Here you have the 3rd carbon dioxide. For 1 molecule of pyruvic acid, we got 3 molecules of carbon dioxide. But as glycolysis ended, we had 2 molecules of pyruvic acid. So with 2 molecules of pyruvic acid, we get 6 molecules of carbon dioxide, right? So this is complete oxidation. Still, we have not yet spoken about where oxygen is utilized. We'll learn about it. I want you to understand, but at this point, complete oxidation has happened. Okay? So, yeah, in the next step, the succinyl coenzyme A, which contains 4 carbons, will be converted to a 4-carbon acid called succinic acid. And while succinyl coenzyme A is getting converted to succinic acid, simultaneously a molecule of GTP will get synthesized. Now this GTP is also an energy-rich molecule like ATP. As soon as GTP is formed, GTP's formation is coupled to ATP formation. This GTP will break down, give that phosphate to ADP and make ATP. Alright, so this is equivalent to ATP molecule. You can think about it like that. In the next step, succinic acid gets converted to another 4-carbon acid called fumaric acid. Fumaric acid further gets converted to malic acid. 4 carbons again. Malic acid will further get converted, or it regenerates oxaloacetic acid. Since the cyclic it continues, you call it Krebs cycle. I want you to remember how during the formation of oxaloacetic acid, another molecule of NADH plus H plus gets formed. NADH was formed in glycolysis, 2 NADH. In link reaction, for 2 pyruvic acid, we get 2 NADH, totally 4. In Krebs cycle, per acetyl-CoA, you have 3 NADH. So from 2 acetyl-CoA, you have 4 NADH formed so far. Alright, so 6 NADH formed so far, 1, 2, 3. From 1 acetyl-CoA, since we'll have 2 acetyl-CoA, in the Krebs cycle alone, we will get 6 NADH formed. Alright, and also another thing that I forgot to mention is, during the formation of fumaric acid to from succinic acid, a molecule of FADH2 will form. FADH2 is short for flavin adenine dinucleotide. H is to show that it is its reduced form. H does not stand for anything. It shows that it is reduced form. Similar to how NADH is an energy-rich molecule capable of giving us 3 ATP each, FADH2 also is an energy-rich molecule that will give us 2 2 ATP each. Every FADH2 molecule is equivalent to roughly 2 ATP molecules. Alright. Even over here, it's important for you to remember where all decarboxylation is happening, where is GTP getting synthesized, where is NADH getting formed, where is coenzyme A getting released. So all of these you should remember. Alright. Now, in aerobic respiration, what will have to happen to pyruvic acid? It will undergo oxidative decarboxylation in the mitochondrial matrix. It will also go through the Krebs cycle in the mitochondrial matrix. So within the mitochondrial matrix, the following reactions will happen in aerobic respiration. So at the end of aerobic respiration, what is the fate of pyruvic acid? Each molecule of pyruvic acid will completely get broken down and release 3 carbon dioxide, 1 carbon dioxide in the link reaction, 1 in the link reaction, okay, 1 in link reaction and 2 in Krebs cycle, right? It will produce 4 NADH, 1 in link reaction, 3 in Krebs cycle, FADH2, 1 in Krebs cycle, ATP, from GTP you get this ATP, this is 1 in Krebs cycle, right? So yeah, this is basically what we get at the end of aerobic respiration. I told you how NADH, FADH2 are high energy molecules. They will give us ATP. NADH will give 3 ATP, FADH2 will give 2 ATP. How exactly does that happen? I told you it's like a check. It has to go into the, to a bank present in the mitochondria, and there it has to convert that check to cash. From NADH you get ATP. So how exactly that happens, we'll have to learn next. Okay. So I'll tell you how. You take a cell, right? You have, let's say this is a plant cell, you have the cell wall, cell membrane and everything. That's not important right now. And within the cytoplasm of this cell, glycolysis happened. Glucose in multiple steps, like 10 steps, form 2 molecules of pyruvic acid, right? Now this pyruvic acid from the cytoplasm was transported into the mitochondrial matrix, right? So in the mitochondrial matrix, pyruvic acid formed acetyl coenzyme A. This is the link reaction. This acetyl CoA entered the Krebs cycle. In the Krebs cycle, what was forming? NADH and FADH2 formed. Now this NADH and FADH2 that was formed in the mitochondrial matrix has to go to a bank, and that bank is located in the inner mitochondrial membrane. In the inner mitochondrial membrane, you have a series of electron transport or electron carriers to which NADH and FADH2 will donate the electrons. And similar to how ATP got synthesized during the light reaction in photosynthesis by chemiosmotic hypothesis, something very similar happens over here as well. That is called oxidative phosphorylation. So by a process of oxidative phosphorylation, NADH and FADH2 will produce ATP inside the mitochondria. Alright, so what exactly happens here? We will understand. That is called the electron transport chain. It is a pathway through which, as the name itself indicates, electrons gets passed on from one electron carrier to another electron carrier and create a proton gradient, and as a result ATP generation will happen. So where does this happen? It happens in the inner mitochondrial membrane, like I already told you. There are 5 complexes present in the inner mitochondrial membrane. Similar to how you saw in a photosystem, you have so many different types of pigments, proteins bound to it, enzymes and all of that. In these complexes also you will have, complex means there's more than one thing there, right? There will be more than one electron carrier present in each complex, and they are involved in electron transport, which will help in the formation of ATP. There are 5 such complexes. So what basically happens in the electron transport chain is a series of reduction oxidation reactions. Electrons are being transferred, means if something is losing an electron, it's oxidation. If something is gaining an electron, it's getting reduced, right? So oxidation reduction reactions keep happening, and energy that is stored in NADH and FADH2 will get converted to ATP, is utilized in the formation of ATP. Okay. So what are these 5 complexes? I will tell you. Each of them, you can call them complex 1, 2, 3, 4, 5, but they also have some fancy names. Complex 1 is called NADH dehydrogenase. Complex 2 is called succinate dehydrogenase. I forgot to mention one thing in the Krebs cycle. See, over here FADH2 is formed, right? This FADH2 formation, succinic acid getting converted to fumaric acid, this is catalyzed by the enzyme called succinate dehydrogenase. In every other step also, you have enzymes catalyzing. Okay. All of these other enzymes required for the other steps, they are present in the mitochondrial matrix itself. But this succinate dehydrogenase is present in the inner mitochondrial membrane. And that is exactly what complex 2 is. Okay. So that is where FADH2 also gets synthesized. Remember this, we will talk about it very shortly. Okay. So this is called succinate dehydrogenase. The third complex is called cytochrome BC1 complex. Remember we had B6F complex in photosynthesis. This is similar. It's a protein electron carrier. It's called cytochrome BC1 complex. Complex 4 is called cytochrome C oxidase. Complex 5 is something that you're already familiar with from photosynthesis. It is called ATP synthase. It will have 2 components, F0 and F1. You know about it, right? And apart from that, there are certain mobile electron carriers that are not present within the membrane. I'm sorry, they are present within the membrane. They are ubiquinone and cytochrome C. Okay. Now, in each complex, there is more than one electron carrier present. In complex 1, NADH dehydrogenase, you have an iron sulfur protein that is represented as FES, and you have something called FMN, which is flavin adenine mononucleotide. It's expansion I've written over here. Alright, both of these components are present in complex 1. In complex 2 also you will have iron sulfur protein. In complex 3 also you will have iron sulfur protein. So this is common to 1, 2 and 3. In complex 2, in addition to iron sulfur protein, you will have flavin adenine dinucleotide. Alright, FAD is flavin adenine dinucleotide. FMN is flavin mononucleotide. This is FAD, dinucleotide. First complex, remember, mono. First means 1, flavin mononucleotide. Second means di 2. That's how you remember it, but FAD is flavin adenine dinucleotide. Third one, FES is constant. What is the complex called? Cytochrome BC1. So in addition to iron sulfur protein, you will have cytochrome B and cytochrome C1. Okay. Fourth complex is cytochrome C oxidase. In the fourth complex, you will have cytochromes as well as some copper centers. You have 2 cytochromes, cytochrome A and cytochrome A3. Along with that, you will have copper center A and copper center B. In ATP synthase, similar to photosynthesis, you will have F0 and F1. Alright, this I hope you remember easily. Next, we will move on. How exactly they are located in the membrane, I will show to you. If you can think of this as the mitochondria inner membrane, this is how the mitochondria is, right? It has a lot of infoldings called cristae. This is the outer membrane, and in here you have the inner membrane. In between the two, there is space that is called intermembrane space. It is over here in the matrix that Krebs cycle and link reaction took place. So whatever NADH and FADH2 are formed, they are present in the matrix, right? It is in the inner membrane that the complex of electron transport chain are present. So this is our ETS. If you look at the electron transport system, in the inner membrane, you have the phospholipid bilayer. In between the phospholipid bilayer, you will have these complexes present. Alright, so you have complex 1, 3, 2, 4, 5 and so on. Okay, all of that will be present within the inner mitochondrial membrane. Now, if you look at this inner mitochondrial membrane, on one side of the inner mitochondrial membrane, you have the matrix towards the inside. Towards the outside, over here, you will have intermembrane space. This is what has been represented in this illustration over here. So what you're looking at here is the inner membrane. It has its phospholipid bilayer. Within that, you have all of these complexes present. Alright, that's complex 1, 2, 3, 4, 5. And each complex has its individual components present. I told you there are two mobile electron carriers. This is one mobile electron carrier, ubiquinone. This is the other electron carrier, mobile electron carrier, cytochrome C. Mobile electron carrier means they can move within the membrane or in the intermembrane space, they can move. That is what mobile means, right? So I want you to notice how on one side of the membrane towards the inside, you have matrix, and towards the outside you have intermembrane space. Okay, so I've also mentioned what are the different components present in each complex. Notice how FES is present in all the three, 1, 2 and 3. It's not in sequence, it's not 1, 2, 3, 4, 5, it's 1, 3, 2, 4, 5. Okay, notice how 1, 2 and 3, all of them have iron sulfur protein. Complex 1 NADH dehydrogenase in addition to iron sulfur protein contains FMN, flavin mononucleotide. Complex 2 in addition to FES contains flavin adenine dinucleotide. Complex 3 in addition to FES contains cytochrome B, cytochrome C1. Complex 4 contains 2 cytochromes and 2 copper centers. So you have copper center A, copper center B, cytochrome A, cytochrome A3, and complex 5 contains an F0 particle present within the membrane with a head F1 particle that is projecting towards the mitochondrial matrix. So this will have the head towards the matrix, so that whatever ATP gets produced, that is released into the mitochondrial matrix. Alright, now let's see how electron transport will take place. First, let's consider NADH. What happens to NADH? NADH was present in the matrix. Now this NADH has a pair of electrons. This NADH will donate or pass on the electrons to NADH dehydrogenase complex and it will get oxidized. In return, this will accept the electron and become reduced, right? So in the NADH dehydrogenase complex, you have many components. You have two components, right? You have FES and FMN. So first the electron is accepted by FMN, then the electron is accepted by FES. Now this electron present in complex 1 is transported not to complex 2 but to complex 3. Remember, whenever NADH, we are talking about the electron transport of electron that we obtain from NADH, complex 2 will not come in the picture. It has 1, 3, 4, that's all, not complex 2. So from complex 1, electron has to be passed on to complex 3. So if electron has to be passed on from one complex to another complex within the membrane, it cannot just throw the electron there because it's a charged particle. It has to happen through an electron carrier. So this ubiquinone which is present within the membrane will pick up this electron from FES. First FMN, then FES, from FES to ubiquinone which is a mobile electron carrier. This mobile electron carrier will catch the electron from this, become reduced to form ubiquinol. UQ is ubiquinone. It accepts the electron and becomes ubiquinone. quinol. Okay, so this ubiquinol will also simultaneously take up a proton from the matrix side and pump it inside the intermembrane space. Okay, so it donates the electron to complex 3. Within complex 3, you have some components. So first, the electron is passed on to cytochrome B, then to iron sulfur protein, then to cytochrome C1. FMN, FES, ubiquinone, cytochrome B, iron sulfur protein, cytochrome C1. Now from the third complex, electron has to be transported to the fourth complex, which is here. Again, transport of electron cannot happen through the membrane because it is charged. You need an electron carrier to carry it from one complex to another. Right now we have another mobile electron carrier which is called cytochrome C. This ubiquinone is located within the inner membrane. This is present within the inner membrane. Look at the position of cytochrome C. Where is it present? Is it present within the inner membrane? Cytochrome C is present on the outer side of inner membrane. So it will accept the electron from complex 3, become reduced and further pass it on to complex 4. In complex 4, you have copper centers, you have cytochromes as well. From cytochrome C, electron is passed on to complex 4 which is called cytochrome C oxidase. Why is it called cytochrome C oxidase? Because it accepts the electron from cytochrome C and makes it oxidized, right? It gets reduced and makes cytochrome C oxidized. So we call it cytochrome C oxidase. Over here, electron is first passed on to copper center A, then to cytochrome A, then to cytochrome A3, then to copper center B. Ultimately, I've been telling you this is an oxygen requiring process. We call it aerobic respiration. Oxygen is required. Where is oxygen required? Oxygen is the final electron acceptor and also it accepts protons from the matrix. Oxygen will take up electrons from copper center B. It will also combine with protons present in the matrix and it will form a molecule of water. Oxygen's role is towards the end of aerobic respiration, but it is very, very essential. If oxidation is very important because it is the final electron acceptor as well as it accepts protons from the matrix. Alright, so that is why oxygen's role is very, very important in aerobic respiration because it's the final electron acceptor. Now I told you from one NADH molecule you get 3 ATPs, right? How exactly can we get 3 ATP from one NADH molecule? We'll understand now. As electrons are being transported through complex 1, 3 and 4, protons from the matrix side are pumped to the intermembrane space. When electrons are being transported through first complex, 4 protons are pumped from the matrix to the intermembrane space. When electron is being transported through complex number 4, wait, 3 also. Yeah, where is 3? 3 is here. 1 is here, 3 is here. When it's being transported through complex number 3, 4 protons are pumped from matrix side to intermembrane space. When electrons are being transported through complex number 4, 2 protons are pumped from matrix side to intermembrane space. So per NADH molecule, we can see 4 plus 4 plus 2, a total of 10 protons are pumped from the matrix side to the intermembrane space. Now that generates a proton gradient. There will be more protons present towards the intermembrane space compared to the matrix side. Now these protons have to escape and go outside. Similar to how ATP synthesis happened in photosynthesis, here also these protons will try to move out through this F1 channel that is present in the membrane. As it moves out, there will be conformational changes happening in the ATP synthase head and ATP formation will take place. According to the updated NCERT, remember in the beginning I told you there is an update in NCERT, right? This is the update that I'm talking about. Earlier they had mentioned it was 2 ATP, right now, 2 protons, right now they have mentioned that it is 4 protons. According to latest update in NCERT, you can see how for formation of 1 ATP, 4 protons are required. Right now we have 10 protons. So from 10 protons, you can roughly get 2.5 ATP. That we will round off to 3 ATP. So NADH gave the electrons because the electrons were pumped, because the electrons were transported, protons were pumped from the matrix side to the intermembrane space. As the protons tried to escape, ATP was synthesized, right? So the electrons ultimately came from NADH. Therefore, we can say that one NADH will give approximately 3 ATP. You have different books giving you different numbers, but this is according to the latest NCERT update. Where did this NADH get its electron from? It got its electron during breakdown of glucose. So ultimately NADH, FADH2 got their electrons during oxidation of glucose. Ultimately all of this ATP is coming from glucose itself, right? So roughly per NADH molecule, you can get 3 ATPs through the electron transport chain. Next, we'll talk about what is the fate of FADH2. FADH2 will not use or will not, complex 1 is not involved in the oxidation of FADH2. FADH2, remember I told you during formation of FADH2 there is succinate dehydrogenase that is present in the membrane, that is this complex 2 here. FADH2 is formed during conversion of succinic acid to fumaric acid. That electron present in FADH2 is immediately passed on to succinate dehydrogenase complex. This succinate dehydrogenase complex will pass on that electron from the second complex to the third complex. In the third complex, same story again. So, where is it? Yeah. So, here is the second complex. If electron has to be passed on from the second to the third complex, again they can't play catch-catch. It will happen through ubiquinone itself. Ubiquinone, the mobile electron carrier, will accept the electron, become ubiquinol, pass on the electron to the third complex, first to cytochrome B, then to iron sulfur protein, then to cytochrome C, C1. So while this electron transport is happening, 4 protons are pumped from the matrix side to the intermembrane space. Again, same story. The electron is passed on to cytochrome C, from cytochrome C to complex 4 and ultimately to oxygen, and a water molecule will form. The story is the same. But the difference here is complex 2 does not pump any proteins. Complex 1 was pumping proteins, right? When it came to NADH, complex 1 was a protein, not protein, I'm saying protein repeatedly, I mean proton, okay? So this is a proton pump. It was pumping protons. But this is not a proton pump, so it does not pump protons. You will get the same 4 protons from complex 3 and the same 2 protons from complex 2. In that case, we are getting a total of 8 protons from one FADH2. Right? We got 8 protons from one FADH2. For formation of 1 ATP, you require 4 protons. Right now, how many do I have? 4 plus 2. I've written 8. Sorry, this is 6. So, from 6 protons, you can roughly get around 1.5 ATP, which we will round off to 2 ATP. Alright, so that's about how electron transport happens. Now, we will have to talk about the respiratory balance sheet. From one molecule of glucose, how much energy, how much ATP are we obtaining? Before we do that, we have to make certain assumptions. They are, firstly, there is a sequential and orderly pathway that is happening in the cell. These are ideal conditions. It doesn't always happen like that. So we should assume that it has happened sequentially in an orderly pathway. Pyruvic acid enters the mitochondrial matrix. It is not used in anywhere else for any other process. And pyruvic acid undergoes oxidative decarboxylation. Acetyl-CoA enters the Krebs cycle. The NADH that was produced in the cytoplasm during glycolysis moves into the mitochondrial matrix to undergo, to participate in the electron transport chain. So these are some assumptions that we have to make. There is a sequential orderly pathway functioning with one substrate forming the next, and with glycolysis TCA ETS following one after the other. Excuse me. Also, whatever NADH is synthesized during glycolysis will have to be transferred to the mitochondria to undergo oxidative phosphorylation in ETS. No intermediates in the pathway, be it glycolysis intermediates or Krebs cycle intermediates, are used up for anything else. It is not withdrawn from the pathways for any other reason, and also only glucose is being respired. No other substrate is being respired. These are some assumptions we have to keep. Assuming all these points, if you calculate how many ATP we can get from aerobic respiration from one molecule of glucose, we will arrive at a number 38. Okay, finally we will get 38 ATP. I'll tell you how. I think I had a slide for that as well. Okay, I don't. We'll calculate. In glycolysis, we got 2 ATP, that is the net gain. Right, we made 4 but 2 was utilized, so net gain of ATP in glycolysis is 2. In the 6th step of glycolysis, we got 2 NADH molecule. If each NADH is equivalent to 3 ATP, then we will get a total of 6 ATP from glycolysis. So from glycolysis alone, you got 8 ATP, right? This is from glycolysis. Talking about pyruvate oxidation, which is also called oxidative decarboxylation or link reaction. 2 NADH are formed for 2 pyruvic acid molecules. Each NADH is equivalent to 3 ATP, so we get a total of 6 ATP from link reaction. In Krebs cycle, we got 1 GTP and 6 NADH molecules, 2 FADH2 molecules, right? For 2 molecules of acetyl-CoA. 2 turns, you get 2 molecules of acetyl coenzyme, you will get a total of 2 FADH2, 2 GTP, 6 NADH, right? So if 2 ATP you get from GTP, 6 NADH you get in 3 different steps. One is during formation of alpha ketogluteric acid, one is during the formation of succinyl-CoA, one is during the regeneration of oxaloacetic acid. So you get 3 per acetyl-CoA. Since we'll have 2 of them, we'll get a total of 6 NADH. Each NADH is equivalent to 3 ATP, so you get a total of 18 ATP from NADH. During the formation of fumaric acid, you will also get succinic acid to fumaric acid, you will get 2 FADH2. Each one is equivalent to 2 ATP, so you get 4 ATP here. So 18 to 20, 24 ATP you will get from Krebs cycle. If you sum it all up, you will get 38 ATP in total. Compare this with how much you got from anaerobic respiration. In anaerobic respiration, all we can get is 2 ATPs. But in aerobic respiration, you get a total of 38 ATPs. Along with that, you get 38 ATPs. Along with that, 6 molecules of carbon dioxide is released because in aerobic respiration, complete oxidation of glucose will take place. Alright, so yeah, that's about the respiratory balance sheet. Let's compare aerobic and anaerobic respiration. Aerobic respiration utilizes oxygen, anaerobic respiration does not utilize oxygen. During aerobic respiration, complete oxidation of glucose will take place. During anaerobic respiration, only partial oxidation of glucose will take place, right? So fermentation, partial oxidation, and aerobic respiration is completely degraded, complete oxidation. Let's talk about how much energy is trapped in ATP. During fermentation, you get only 2 molecules of ATP. During aerobic respiration, you get much higher, which is roughly around 38 ATPs under ideal conditions. Okay. NADH oxidation, NADH going to the ETS and getting oxidized, that will happen much rapidly in aerobic respiration, and it's slow in fermentation. Alright, these are some differences. Right now I want to highlight 3 different types of ATP formation. We call formation of ATP where ADP, 2 ADP of phosphate groups gets added and forms ATP. That process is called phosphorylation. There are 3 types of phosphorylation: photophosphorylation, substrate level phosphorylation, oxidative phosphorylation. We've learnt about photophosphorylation in photosynthesis by utilizing sunlight, energy in the sunlight, how ATP gets synthesized during light reaction, that is photophosphorylation. Substrate level phosphorylation we have seen in glycolysis, where ATP gets synthesized here. Where is glycolysis? Okay. So this ATP synthesis here and here. All right, wait. Yeah. This and this is substrate level phosphorylation. Photophosphorylation happens in photosynthesis. In Krebs cycle, what we saw, that is also substrate level phosphorylation, where phosphate from a substrate is removed and given to another molecule, ADP, where ATP formation will happen, right? That is substrate level phosphorylation. Over here you can see how a phosphate was already present in 1,3 bisphosphoglyceric acid. That phosphate from this was removed and added to ADP to get ATP. If that is happening, then you call it substrate level phosphorylation. If ATP synthesis happens in the inner mitochondrial membrane because of a series of oxidation reduction reactions, then you call it oxidative phosphorylation. Whatever ATP is synthesized here because of NADH, FADH2, this is called oxidative phosphorylation, where what happens you have to remember. Okay? All right. That's about the difference between photophosphorylation, substrate level phosphorylation and oxidative phosphorylation. Now we have almost reached the end of the chapter. We have to learn about why respiration is called an amphibolic pathway. Amphi means both. Okay, generally we used to consider photosynthesis as an anabolic process which is involved in building up of glucose, and respiration as a catabolic process which is a breaking down reaction, right? Glucose is getting broken down. So traditionally the belief is that respiration is a catabolic process. But here they say that respiration is both anabolic and catabolic. How do we explain that? Now, when carbohydrates are broken down, glycolysis begins with glucose. Starting point is glucose in glycolysis. I told you that during certain conditions, fats and proteins also can be used as respiratory substrates. If molecules other than carbohydrates are used as respiratory substrates, then they don't enter the cycle as glucose. They will enter somewhere in between. For example, fats are broken down into glycerol and fatty acids. Glycerol can enter the pathway as glyceraldehyde 3-phosphate. Glycerol to glyceraldehyde and then enter the pathway. Fatty acids can enter the pathway after getting converted into acetyl-CoA. Proteins will first get degraded or broken down to amino acids. Amino acids will undergo deamination. Later, they can enter the pathway either at the point of acetyl-CoA, either as pyruvic acid or some other intermediate during the Krebs cycle. In different routes, it can enter the respiratory pathway. Okay. Now, if a cell requires proteins, amino acids have to form, amino acids have to join, right? At that time, the pyruvic acid that is produced here may not enter the mitochondrial matrix. It may be withdrawn from this pathway and utilized for the synthesis of proteins. If it enters the mitochondrial matrix, it will get broken down. But this same intermediate can be withdrawn from the pathway and used to synthesize something. Whenever something is getting synthesized, what do we call it? We call it anabolism. This acetyl-CoA can be withdrawn from the cycle and used in the synthesis of fatty acids. Glyceraldehyde can be withdrawn from the cycle and used in the synthesis of glycerol or probably fats. So whatever intermediates you have in the respiratory pathway, they can be withdrawn and utilized to synthesize some things as well, some molecules as well. So you can consider this to be anabolic as well, because it is involved in some intermediates can be withdrawn and involved in the synthesis of some other bigger molecules as well. So we call it an amphibolic pathway. All right, now let's move to the last part of the chapter, which is respiratory quotient. Respiratory quotient is abbreviated as RQ. It is also called the respiratory ratio sometimes. Okay. So how much RQ is depends on what is the substrate that is being respired. What is respiratory quotient? Respiratory quotient is basically the ratio of how much carbon dioxide was released during respiration divided by how much oxygen was utilized for this process. Okay, this is only for aerobic respiration. How much carbon dioxide, how many molecules of carbon dioxide, or what is the volume of carbon dioxide that was released over what is the volume of oxygen that was utilized. All right, so it is a ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed. For example, if you see, I told you it depends on the substrate, right? So if the substrate is glucose, then 6 molecules of carbon dioxide is evolved, 6 molecules of oxygen is utilized or consumed. So 6 divided by 6 is 1. For glucose or carbohydrates, you can say that the RQ value is 1. If you consider tripalmitin as the respiratory substrate, then in tripalmitin, 102 molecules of carbon dioxide is evolved, 145 molecules of oxygen is consumed. So 102 divided by 145 will give us an RQ value of 0.7. Alright, so that is basically the respiratory quotient. It is, I think, around 0.9 for proteins, and for some organic acids it can be more than 1. Alright, so that's about respiratory quotient. With that, we've come to the end of the chapter. I hope this was helpful. If you have any doubts, please feel free to drop it in the comments. I will answer your queries as soon as I can. Thank you. I will see you in the next class with another chapter, Plant Growth and Development. Keep staying focused and study hard. All the very best. Thank you.